Okay. Good afternoon, everyone. We're going to get the session rolling at this point. Our first speaker is Delaney Long, who will be speaking to us about parasites, striped bass in the Hudson River. And I'm Fred Henson. I will be your moderator for the afternoon. And my co-moderator, Patrick, is right in front of me. So with that, Delaney, the floor is yours.
Thank you. Hello, everyone. Can you hear me okay?
Yes.
Cool. So my name is Delaney, and in collaboration with my advisor, Dr. Florian Reyda of the Oneonta Biological Field Station, and Jessica Best of the New York State DEC, I did a survey on the parasite community of the striped bass in the Hudson River. To get started, I know everyone in this room knows about the Hudson River already, but I wanted to give a brief explanation. It was originally used by the Lenape and Munsee tribes, and they called it the Mahanitalk, which translates to the river that flows both ways. Then Henry Hudson discovered the river. It was used in the growth of the Erie Canal and industrialization in New York State, and that led to a lot of pollutions, which led to restoration efforts and acts such as the Clean Water Act, as well as other government policies keeping our waters protected. Striped bass are one of the main predators of this Hudson River ecosystem, and therefore they're excellent hosts of parasites. They're an anadromous species, which means that they spend most of their life in the marine system, and they go to the freshwater system, such as the Hudson River, to reproduce. They're really recreational. They're popular for recreational fishing, as well as an economically important species, as I'm sure you all know. But due to their popularity, they are facing a population decline. So this is just a little food web of the Hudson River. You know, you have your bacteria, phytoplankton, and then a little bit higher you have your zooplankton, your copepods, you have some midges, amphipods, and then your more charismatic fish, as well as a bird and a person, which are all integral parts of the ecosystem. But I would argue this is not a full picture. There's parasite communities and parasite food webs circling throughout all of these communities that are really paid little attention to. So what did I do? My survey work, as I said, was with Jess and Dr. Reyda, going down to the New Paltz office in DEC to actually do all the measurements, but we went to 5 different sites along the Hudson River to collect striped bass. We got about 20 fish from each site, sampling twice, 2 separate times. At the New Paltz office, the fish were weighed, measured, sexed, there was fillet samples taken, spleen samples taken, and scale samples taken. That was all for the New Paltz DEC's PCB work, which I'm sure Jess will talk more about at some point, maybe. Not today, sorry, never mind. Not today, I forget I said that. But I'll talk to you more about that if you're interested. That is not what I'm focused on today. I'm focused on the parasite survey that I did. So after Jess's crew at New Paltz did all of their work, I swung in and I cut out their organs and I put them in a bag and I drove them back to my lab. And then while I was in my lab, I looked through everything I found. I ID'd my parasites, made slides, and did some other tests, which I'll get into in just a second. Before I do that, I want to talk about the roles of parasites in the perspective of a fish host. As fisheries experts, I think this is important knowledge to have. So there's something called a definitive host, which is when the parasite reaches its mature form, its final destination, if you will. So it's able to reproduce, it's fully mature, and it's going to reproduce in the intestines of the fish, and it's then going to be passed as eggs or larval stages in with the fish feces to complete their life cycle once again. There's also an intermediate host. This would be an example of a parasite being in a body cavity or organs that are outside of the digestive tract. This parasite, if it's in any of these places, it's not at its final host yet. It's got some more steps along the food web to travel. And so if a bigger fish or a bird eats this fish that has a worm in its body cavity, it's then going to make it to the intestines of that definitive host, the bird definitive host, for example. We also have direct life cycles. This is when a parasite is usually external on the gills or the fins of fish, and it doesn't have another host, it doesn't have to make it anywhere else. It can reproduce and reinfect the fish host or a fish of the same species. In this example, I have it on the gills and the fins. So what do we care, why do we care about parasites in fisheries, right? That's the big question. They can be used as indicators. So I can tell about population dynamics, community structure, environmental stress, food web structure, and biodiversity based off of the parasite composition from a population. So there was a 1976 survey done of striped bass parasites in the Chesapeake Bay. And this was my first introduction into the parasites of the striped bass. You know, before fieldwork had started, I still had light in my eyes and was excited about being a graduate student. I was so excited. They found 45 different species of parasites. They found 3 kinds of viruses, 1 kind of bacteria, and 11 kind of protozoa. I did not look for these kinds of infections on the striped bass, so I want to mention that they did it, but it wasn't what I was looking for, so I can't compare my findings to this. What they did find is 3 species of monoginian, which are characterized by an indirect life cycle, as well as a attachment organ called the hapter. They found 9 species of diginian worms, which are characterized by having those 2 suckers that you see, as well as an asexual stage and a sexual stage of reproduction. They found 4 species of cestodes, or tapeworms as you may more commonly hear them referred to as. These are segmented worms with a skull-X head. They found 1 species of acanthocephalin, which are characterized as the thorny-headed worms. You can see the proboscis in the picture, that's the thorny head. They found 4 species of nematodes, which are a tube within a tube. They have a cuticle layer as well as a pseudocoel inner layer. They found 6 species of crustacean, parasitic crustacean, like copepods, which are the direct life cycles that attach to gills. They found 1 species of leech, which I'm sure you're all familiar with what a leech is. And they found 1 species of Glaucidia. Oh, sorry. Okay, 1 species of Glaucidia, which is the parasitic stage of a freshwater mussel. So I was really excited about that. I was pumped. I couldn't wait to find 45 different species of parasites in my fish. I spent 10 weeks traveling a long way to get parasites, to get fish, and I found 5 species of worms in 149 fish. So I found 1 species of monogene, 1 species of trematode, 1 species of acanthocephala, 1 species of nematode, and 1 species of parasitic crustacean. I was a little disappointed, I'm not going to lie. Just to walk you through what I did find, and if I found 45 parasites, I couldn't tell you about all I found. I wouldn't have enough time, so that's the bonus. The monogene that I found is called Gammacella macroura. Also, parasites don't tend to have fun common names because people don't care about them enough. I do. So in Gammacella macroura, our monogene, they are an indirect life cycle, so they just stay at one species their whole life. So they can reproduce on the striped bass, they can reinfect the striped bass, they can infect another striped bass. They're infected in euryhaline water, so the Hudson River in that transition zone is a really perfect breeding ground for them. They can cause microscopic lesions on gills, gill pallor, tissue injury from the attachment of the suckers, and they decrease the white blood cell count of the fish host. So they're obviously pretty harmful. They're more harmful in hatchery situations just because they don't use the food web to spread around, they use the population of the fish species to spread around. Next, I found Stephanostomum tenue, which I'll talk a little bit more about later in my presentation, but for now they are, also sorry, the formatting changed. You could see all the words on my computer. In Stephanostomum tenue, it starts in that eastern mud snail, which is that then the worms go to the Atlantic silver side, and the final host is the striped bass. That's where they reach maturity. However, if an American eel or a rainbow trout were to eat an Atlantic silver side that had juvenile stages of the trematodes of the worm species, they could get infected. Now, the worm won't become sexually mature in those fish, but it can still cause mass casualties and other pathology problems. These are marine transmitted, but they are very understudied species, so we don't know much about this type of worm. Next, I found Pomphorhynchus rocae, an acanthocephalin species. You can see those lesions on the, those are the outside of the intestines on the top picture. So that is from the proboscis of the worm embedding into the intestine. You can see some pretty serious pathology on the intestines alone, and that's what they look like on the inside. Usually parasites aren't fun colors, so these ones are really excited to find. They're marine transmitted, and they start in a marine amphipod, and we're not sure if they go from we believe they go to a white catfish and then the striped bass, but we're not entirely sure. Again, a lot of these species are highly understudied, so we don't have too much information about them. As I said, they cause lesions on the intestines, but they also it's been shown that they can uptake heavy metals. So fish will have really low concentrations of heavy metals that have high infections of this species, compared to fish that don't have infections with this species. And there is also some seasonality displayed, so over the summer we'll find a lot more than we would this time of year, for example. Next, I have Philomitra rubra, and the video's not going to play, but I have worms squirming around in the petri dish. I thought that'd be cool. If you want to see it, come talk to me after. They are a freshwater transmitted species, and they've also been known to cause mass deaths in nurseries. These worms are free living in the body cavity, so once we take the fillet sample of the fish, you can just see these guys wiggling around. Anyone that was in that room got really freaked out by it, they can tell you. They cause some severe visceral edema and granuloma in the body cavity. And again, understudied, not a fully understood life cycle. We believe they go from a free-living copepod, different than a parasitic copepod, to the striped bass directly, but there could be another fish as an intermediate host. And lastly, I was going to talk about my copepod that I found, Ergasilus librasis. That's a picture of them connected to the gills, and they use those little hooks that they have on their head to attach to the gills. These are marine transmitted and a bit more studied than the rest of the species we're talking about, just because of how easily they are to find on the striped bass. They can cause gill hyperplasia as well as an increased number of mucus cells on the gills, causing to suffocation even when the fish are in otherwise ideal environments. And as I said, they're reported on very high amounts all over the East Coast on the striped bass with peak seasonality from May to June. So here's a little bit about my results. I found the Ergasilus librasis, this is the prevalence. So this is the, this is how many of the fish, the prevalence is the amount of fish infected. No, the amount of fish infected, sorry. So we found the copepod, the Ergasilus librasis, at all sites at pretty even ratios, except for that Troy spot, which is a little unusual because it's a marine transmitted species, so we kind of expected it to go down the further up the river we got, but we saw the opposite. We have Gammacella macroaura, our monogene. We only found that species in Troy. However, that's by far the smallest one we have. It's very hard to find within the gills, so it's possible we missed it. We did only find 3 of those in Troy. So, Philomitra rubra, our nematode species, we saw pretty consistently. Same with Pomphorhynchus rocae, and Stephanostomum tenyu had an increase in catskill, and otherwise it stayed consistent. And then this is the mean intensity, so the number of parasites per infection on average, split between the species themselves and the different sites. So I have not finished all my data collections yet. We're going to do some more sampling, more pointed sampling this coming spring, but the results that we have so far show us that there is changes happening. There's a decrease in diversity, and we're seeing less species and less amounts of species. But what about what we didn't find, right? There was 45 species found, and we found 5. One of them, and I'm not going to take too much time to go over these, but one of them is Lepocridium setaphoretes. They come from the same snail host as Stephanostomum tenyu, and if a chain is broken in the food web, that could explain their absence, but there's also evidence that some species of parasites just can't cohabitate with other species of parasites. So they can't live in the same snail, despite the fact that they both use that snail to get to their next host. So there's a couple different ecological explanations of why we didn't see that worm. And then there's Homolometron pallidum, which is in a different marine snail, goes to a clam, a polychaete worm, and then Morone fish species. This could be explained by the invasion of invasive clams and mussels, kind of taking over the native clams and mussels, and us not having the native clams anymore to continue that food web. So if one chain in the food web is broken, everything falls apart. You know, it can't keep going. So I have some next steps, which are connecting this problem to the rest of my thesis. And I'm going to make connections with DNA. We have these cercarial stages, we have these adult stages that we found in the striped bass. And even though they match the description of Stephanostomum tenyu, and they've been confirmed by family experts to be Stephanostomum tenyu, the sizes aren't matching up with what's described so far of the worm. So they've all been about double the size of any described species of this worm. So we're going to be finding cercarial stages, which are like larval stages of the worm, to compare with DNA to the adult worms. And I like this slide. It kind of gives you the example of a metacercaria stage is like a baby worm, but you can still tell what it is, and the cercaria stage, you can't match that to a species. So that's what my work will be continuing throughout this semester. Here's my works cited. I want to thank my lab, my parents, everyone that's helped me. I have my cat on here too. She helped. And with that, any questions? Yeah.
With past parasites that you've been comparing the sizes to, have they been found in the same species that you found in the
Yeah, so when you describe a new parasite species, you have to write a whole description paper, and you have to provide measurements, and it's also common practice to provide what's called a syntype or a holotype, which is a slide, a mounted slide of the parasite you found. So that way, if 100 years from now, someone is like, oh, I think I found this worm, but I'm not sure, let me get the original description and see if it matches. We're able to do that. And we're not finding anything that matches, so that's kind of the problem we're having. Any other questions? Yeah.
So, I mean, that's a lot of samples and a lot of dissections and stuff.
Yeah.
How long do these parasites live outside of a living host? And I guess with that as well, when you're processing, how are you like going about trying to find them? Like, are you staining things? Or are you just
Yeah, that's a
taking
good
things
question.
out?
So in terms of how long they stay alive, we made it through all the fish we got within 48 hours of the fish being euthanized, and we never had dead worms. They can survive quite a while. You know, if you froze them and left them for a couple months, you could still find them, but they won't be wriggling around, which that makes them just easier to find, right? And the way we find them, we actually don't stain them. We use just stereoscopes, and we cut through the intestines. We kind of unroll them and open them up, and we just use some lights and a dissecting scope and a lot of caffeine. And we find them, and then we stain them to make the actual slides. So those pictures you saw of the worms that I found, they're not purple when you find them in the intestines, right? I wish that would make my life a lot easier. But no, we stain them and clear them to make them, and their external as well, their internal anatomy visible.
All right, thank you.
Thank you.
Thank you.